that excellent introductory presentation. And just to note on that last point, I think, Jason, you mentioned the experiment in Cornwall. In fact, that experiment has been withdrawn because of the, well, for a number of reasons, I think, but there are some experimental results that came from that, but I think they were somewhat inconclusive because of the lack of a good baseline. And I wonder whether that's something that you might have some views on. I mean, if we are going to be able to study the impacts of these kinds of interventions, how important is a baseline, and what kind of length of time for a baseline of environmental conditions would you recommend in order to be able to detect the implications of some kind of intervention of alkalinity enhancement or anything else? Thank you.
Thank you. So, Professor, when you are ready to answer. Professor, you may need to raise a hand again to be online. Okay, yeah.
My apologies. Okay, thanks, David from Greenpeace for that question. I didn't know that the experiment of Cornwall had ceased, but the fact that their conclusions are uncertain due to a lack of baseline doesn't surprise me. I actually think that the best way to find out what the effects of enhanced ocean alkalinity would be is to go to places that are naturally like that. So, for example, just like we've been studying the effects of increased CO2, you can go to places that have got enhanced alkalinity for perfectly natural reasons. So I'm thinking of coastlines that are karstic, that have got limestone, and rivers entering the water that have got high levels of calcium carbonate in them. Those are perfect natural analogues for looking at the effects of increased alkalinity. You can use gradients going away from these areas with high alkalinity to see at what stages the biology of the system changes without having to interfere with the system. And the beauty of that is that these areas have had high alkalinity for hundreds and thousands, in some cases millions of years. So you can look at the long-term effects of increased alkalinity rather than just short-term shock type experiments where you can suddenly see changes in the phytoplankton, for example. So that would be my best approach, I think, for looking at how the ocean responds to increased alkalinity.
Okay. I think we can have one more question. Canada?
Thank you very much for the interesting presentation. I just have a follow-up question to what was brought up about these experiments that are happening from sewage outfalls or similar outfalls on the coastlines. Would you have any thoughts about the transferability of results or research happening from outfalls to perhaps larger activities that could happen at sea? Would there be anything in that type of research from outfalls that would be particularly relevant to looking at how ocean alkalinity enhancement would perform in the open ocean? Thank you.
Thank you, Professor.
Well, clearly the near shore and the open ocean are completely different systems, especially because we are naturally producing sewage, or unnaturally producing sewage that affects the coastal ecosystems in a big way, especially in terms of organic enrichment and also inorganic enrichment with nitrates and phosphates. So the coastal system reacts in a completely different way these days as to the open ocean, which is much more oligotrophic, that isn't so saturated in nitrogen and so on. So the biological differences you notice when you change the alkalinity of the near shore will certainly be different to what happens when you change the alkalinity offshore on the global ocean. I hope that answers the question. If not, do email me.
Okay, thank you. And we still have another question from Ocean Care.
Thank you very much for that talk, Professor Hall-Spencer, and I very much agree with your assessment of the capacity of carbon dioxide removal that you made at the beginning. But I'd like to make a comment with a question that, we know that coral reefs thrive in stable and narrow environmental conditions, and I find this claim of co-benefits of ocean alkalinity enhancement rather spurious, because we know also that coral reefs exist in hydrodynamic conditions that tend to involve strong mixing, tides, wave action, something that is very difficult to model and to predict. And I would therefore think that precise dosing that you would need to do to ameliorate the risk of ocean acidification would be incredibly difficult in practice. I wondered if you could comment on that. Thank you.
Thank you, Professor.
Well, I couldn't agree more. If you're trying to look after a coral reef in a fish tank, that might be doable because you can carefully monitor the exact doses of alkalinity required. But to do it in a lagoon might be feasible, but very, very expensive. But like I said, if you had a hotel next to a coral reef that you wanted to protect, that might be feasible. But to do it on the scale of, for example, the Red Sea or the Great Barrier Reef would be impossible. And anyway, you'd need to have so much alkalinity that would involve mining, for example, the whole of the Dolomites out to change the ocean chemistry on the scale needed to combat human CO2 emissions. So it's not really going to work, basically.
Okay, thank you. I see no more questions. So I thank you, Professor Hall-Spencer, for sharing your fascinating research results. I thank you very much again. I am very sorry, but because of the technical issue, we have to change the order of the presentation. So our next speaker is in person, a presentation by Dr. Andrea Gaion. Dr. Gaion is a researcher at the Italian Institute for Environmental Protection and Research. His work focuses on biological testing methodologies and the interaction between climate change and contaminants in marine ecosystems. He is actively involved in working groups related to marine environmental science, climate adaptation, and public education. He began his career at ISPRA, later taught in the UK, and returned to the ISPRA. His research particularly addresses the ecotoxicological effects of metals in a naturally enriched marine environment. The title of the presentation is Climate Change and Ocean Acidification Amplify Metal Toxicity and Drive Biological Stress in Mediterranean Mussel. Yeah, Dr. Gaion, when you are ready.
Thank you very much, Chair. I think I requested to share my screen. So, yeah, perfect. So thank you very much, and thank you for giving me this opportunity to present the results of this important work. I also want to thank Professor Hall-Spencer, finally music for my ears, and and most of the results that Professor showed before will be reconfirmed in my presentation as well, so that's a good sign. First of all, let me spend a few words on the role of my institute in Italy. We've always tried to bridge the gap between science, applied science, and legislation. So what we want to do is to conduct research, scientific research, aimed to overcome the limitation set in the normal legislation and improve this process through the years. We are particularly focused on coastal ecosystems and specifically to sediment dredging. So our interest is in each step of the process, so from the sample collection to the sample preparation and handling down to the biological analysis in this case. And we focus specifically on identifying which are the most relevant classes of contaminants for a specific response, or which are the most appropriate or sensitive, or also inappropriate or inaccurate biological testing. And we try to identify as much as possible the confounding factors in biological analysis so we can take them into account in our results. So there's a wealth of knowledge now on climate change effects, especially in the Mediterranean Sea. As was mentioned before, the Mediterranean is a hotspot for climate change. Information on ocean warming, sea warming, ocean acidification, and deoxygenations are well known in the Med, as everywhere in the planet, as well as the marine heat wave events, which our sea really suffers from. We also have a good experience, a good wealth of knowledge on the contaminant effects. So since the 70s, more or less, we started to develop, to accumulate a lot of information about what are the toxicological effects of the legacy contaminants, so metals, the organic molecules that we normally use in life, and we correlated the concentration of these contaminants to the biological stress. So what we have to do now is to combine these two set of data. So we need to understand how the changing of the environment will affect the fate, the transport, and ultimately the toxicity of the contaminants. Most of the studies in this field are coming from lab experiments or mesocosm, just because, as was mentioned before, you can control all the parameters, but by definition, they are unable to recreate the complexity of the natural environment. So long-term field data sets are quite scarce, but that's exactly what we need to understand what's happening in the real world. So the first experiment that we did in this sense, just a flashback to 3 years ago, for those of you who were not present here, we assessed the increased sensitivity of the sea urchin larvae to copper using our 20-year-long data set. So we used the EC50 value as a proxy for the reproductive health of the sea urchin. EC50 is the concentration needed to cause malformation to 50% of a population of larvae. And we noticed a negative trend, especially after 2016. So we argued that the population probably accumulated a lot of stress during the precedent years and reached a tipping point after which was unable to recover. We conducted a first casual analysis, and we identified that the pH and the CO2 affected the response in the EC50 up to 1 year before the sampling. So the effect of the alteration of these parameters had an effect in our measurements up to 1 year later. We then decided to pass to the next level, so we wanted to increase the complexity of our study, and we applied a similar methodology, just improved, to another location. It's a regasification terminal located offshore of the coast of Italy, 12 nautical miles, in between Tuscany and the Gorgona Island. If you've never been there, it's beautiful, so I suggest you to visit it. And this is just a representation of the regasification terminal. So it's nothing but a ship anchored to the seabed, receives the liquid gas, and uses water as a heat exchange to regasify the gas and send it to the national network. So only part of the monitoring program set by our institute to the regasification company included the study of bioaccumulation of metals in mussels and correlating the bioaccumulation to the biological response measured with 4 biomarkers. For those of you who are not confident with this topic, the biomarker is an objective and measurable biological response that acts as an indicator of normal or biological stress. In our experiment, we analyzed 4 biomarkers. The first one is representative of genetic damage. So it's called Comet assay, and the result is represented as a comet. That's the name of the test. And the percentage of DNA in the tail represent the damaged DNA. So the bigger the tail, the more damage is the DNA. The second biomarker is the neutral red retention time, which is a test that is used to verify the stability of cell membranes. We use the membranes of lysosomes, which are very similar to the cell membranes, and by counting the time needed for the lysosome to release the dye into the cytoplasm, we can assess the degree of the stability of the membranes. The third one is simply the hemocyte count, so the equivalent of our blood cells for muscles. And the fourth biomarker is a scale of integrity of the gill tissue of the muscles. We then combine all these four information into one single biological stress index. We didn't want to assign different weights to each biomarker because we didn't want to discriminate one over another or saying that one was important than the other. So it's just a general indication of the stress of the animal, measured with different physiological aspects. Because we had to deal with a lot of numbers, we wanted to set a clear and precise statistical approach from the very beginning. So the first step was to collect the data. So we collected all the biomarkers and heavy metals measurements, downloaded temperature, pH, and oxygen from the Copernicus satellite, daily series for 10 years at the same location. So that brought us to 6,800 readings. And we validated the satellite readings with CTD profiles. And again, because it's a vertical profile in the entire column, we got 68,000 readings for the CTD to use as a validation strategy. And we also identified 19 marine heat wave events in the area. The second step was to clean up the background noise. So what we wanted to do is to normalize the baseline. So how Mussel Watch works is that basically we collect the mussels from a mussel farm. We conduct the baseline analysis, bioaccumulation and biomarkers. We then transplant the mussel in the site that we want to test, or in the control site, as in this case. And we measure the same parameters after 30 or 40 days of exposure. So we wanted to take into account the natural variability of the T0, so the baseline level of the mussels. And that's where we decided to use an innovative approach. So rather than going online and finding a specific method used before in a similar situation, we identified all the different methods used before, 6 of them, and we tested 4 different combinations for each one of these 6 methods, and we identified the best performance in terms of statistical reliability. And using this normalization, we basically eliminated the background noise. Then we did a quality control on the data, so we only collected the data which full information, so 4 on 4 biomarkers, 12 metals, and all the climate covariates. That brought us to 801 information individuals. And then we divided this set of data in 2. The first subset, 70%, were used to run the models and to train the system to run the models. And we used the second subset, 30%, as a validation subset. So we applied the models that we calculated with the first one to test if it was working on the 30% of the data, and it worked. So that's the good news. Then we moved to the next step. So we identified the major factors contributing to the variance response in mussels, and we identified climate change, metal pollution, the effect of the industrial site, the seasonal cycle, and an unknown component, which is basically genetic diversity or anything that can be controlled, like the handling of the mussel before the transplant. We don't know what happened in the mussel farm before, so we wanted to account for this unknown component. And again, we use a similar approach as before to calculate the contribution of this factor. We used 5 different advanced statistical approaches just to see if they were giving the same results, and they did again, so that's another good result for us. So we found the primary stressor, which in our case was climate. In particular, the pH amplifies the metal toxicity we measured in mussels. And then the final step was to project the stress that we measured into the future IPCC scenarios. So let's have a look at some charts. That's what we like after all the stats. So our data confirm the Mediterranean being a hotspot for climate change. So the warming was faster than the global ocean average, brought us to a cumulative increase in temperature of about 0.4 degrees in a decade. We identified 19 marine heat waves, in particular increasing in frequency and intensity after 2008. I just wanted to point out this heat wave in 2022, 5.8 degrees average above the climatological baseline for 23 days. So that's an intense marine heat wave. Similarly, the ocean acidification registered a decrease in the pH, so an increase in the acidity, again faster than the global average. And the deoxygenation was in line with the global average of the ocean. We then conducted a comparison between the two sites, the control site, the Gorgona Island, which is a marine protected area, and the terminal site in terms of bioaccumulation of contaminants. And we didn't see any difference between the two sites, nor between the years. So we can say that basically there was no significant bioaccumulation of contaminant registered in the data set. So we calculated the biological stress index for both sites, and we noticed a positive trend over the decade, which was not statistically as different from the two sites, the control and the exposure site. That means that the stress was similar because of the basin in which the two sites are contained, not because of the exposure of the terminal. So we conducted a causal analysis again, and we identified that 64% of the stress was linked to an indirect pathway. So climate change will affect the bioavailability or the effect of the metals, and that results in an increased biomarker. And 36% was direct stress caused from the climate to the biomarkers. So really the climate stressing the animals directly without any interference of the metals. This is a hopefully clear representation of a very difficult thing. So we wanted to show you the intermediate pathway where ocean acidification increases the bioavailability of metals because increases the percentage of the ionic form of the metal, so it's more bioavailable and will cause more toxicity into the four biomarkers. In particular, the bootstrap support for these was 97%. So that means that if you run 10,000 random permutation, 9,700 times, you have the same results. So it's quite a robust result. And the second one, there was an influence of the temperature on the metal as well, but probably that's because the temperature affected the physiology of the organism, so the organism wasn't unable to fight, in a sense, the bioaccumulation of contaminants. So there was also a component of the temperature, but predominantly it was caused by the acidification. So that's the key result that we obtain. As you can see in the error bars in each factor, all the models really agree a lot on partitioning the variance measured in the stress of the biomarkers. The first stressor is climate, accounting for about 35%. The second stressor is metals, of course, 27%. The effect of the terminal was quantified as 15%. Mainly due to the cold water and chlorinated water emitted by the ship. 4% of the seasonal variability linked with the stress, that's mainly with the gametogenesis of the animals, and about 17% of an unknown component. So that's the projection of the BSI in the future scenarios set by the IPCC. Again, we didn't want to use just one single method, but what we did was to project 50% of the trend that we measured and 50% of the hierarchical GAMS statistical projection, and we corrected that with the three scenarios. So before talking about the scenario, I just wanted to highlight this record high of the BSI. That's that value is in correspondence of the marine heat wave we saw before. So that's very similar to a worst case scenario manifesting today. So we don't have to wait till the worst case scenario projection, but we are already seeing a BSI very close to 70%. We set 70% as a sort of a threshold after which the population of invertebrates could be in a chronic stress state. So the three scenarios, depending on which one you analyze, the low emission scenario, I think unrealistic one, unfortunately. The moderate scenario, the one I'm hoping for, really hoping for, but I don't know. And the high emission scenario, the one that I'm scared of. And between the moderate and the high emission scenario, which is basically where we are today, the chronic stress threshold could be reached in 2035, 2045, so before mid-century. So what are the implications, the practical implications of these results? Well, first of all, we can misunderstand the results of our monitoring programs we had so far. So if we use the standard correlation, more metals, more toxicity, we may argue that the site number 1 has the highest bioaccumulation of metals, and the site number 2 has a lower bioaccumulation of metals. But maybe when we account for the climate, the site number 1 is the one more stressed by the climate, and actually the bioaccumulation of metal is more prevalent in site number 2. So that's a very simple example of how, if we don't account for climate change from now on, we can really misinterpret our results. The second implication is, of course, relevant, very relevant, I think, for us. So all the marine geoengineering techniques, especially the ones which actively input some substances in the ocean, like the ocean ion fertilization. I had a reconfirmation of my worrying because we've just published a paper recently where we assessed the toxicity of iron sulfate because there were not really ecotoxicological information online on this component, not for invertebrate species, and we compare the result that we obtained to the copper. So even if iron sulfate is less toxic than copper, luckily, so we need more iron sulfate to cause 50% of damage in a population, but we are still in the same order of magnitude, so in the tens of ppms. And copper is used as a reference toxicant. In fact, it's used in the antifouling paints. So the toxicity demonstrated by this compound is not so far away from the one of copper. So the key messages I just wanted to pass today are, the first one is that climate change is already affecting the toxicity of contaminants as we know so far, mainly for metals through the pathway of the ocean acidification. We use the Mediterranean mussels, but we can, as a model organism, but we can argue that maybe all the invertebrates can suffer from similar stress. Normally, invertebrates don't have very complex pathways of detoxification processes, so they are very exposed to the variability of the toxicological properties of chemicals because of climate change. The third one is that, as a consequence, we have to update everything that we've known so far. So that means that 50 years of ecotoxicology needs to be revisited and updated taking into account what is happening now to the environment and what is projected to happen in the near future. As you may have noticed in my last interventions, I think it's fundamental to include climate change in every monitoring activity, including the assessment frameworks that we are working on. And this is just an appeal to everyone to share their information on possible long-term data sets that you may have, ideally linked with the authorized industrial activities, because normally the method is always constant for a long period of time. So that would be the ideal data set where the analysis don't change for a very long time. The species are always the same. The location is always the same. So that could really help to understand what's happening and if our results can be confirmed in other places of the planet, with other species, ideally. So finally, we just started to scratch the surface of this unknown world of interaction between climate change and ecotoxicology. There's still a lot that we don't know. There's still a lot to discover. The urgency of this work is set in the figure that I think I show you today, so some alarming figures. So we really need to work hard in this topic. Before concluding, I just want to thank everyone involved in this project. As you can imagine, it was a massive project with a lot of people working on it. So I just want to thank everyone who worked on this project. And I'm available for any question, either here or via email if we don't have time. Thank you very much.
Thank you very much, Dr. Guyon.
Thank you.
A very interesting presentation. I think we can have time for questions or comments. Greenpeace.
Oh, that's good.
Thank you, Chair, and thank you, Andrea, for a very impressive presentation. That's a phenomenal amount of work that you've done. And although you say there's a lot of people involved, there weren't that many names there, so I guess you've been extremely busy. But the approach that you've taken looked very robust, very thorough, and it's good to see that kind of work ongoing. I just had one question on the distinction between climate change and ocean acidification, and whether it's possible for you to say anything more about the distinction between the two, given that to understand whether the increased effect of the metals is something that is linked to the warming effect of CO2 emissions, as in climate change, or the acidifying effects, which are kind of through a different mechanism in a sense. So when we look at incorporating climate change as a concept in all the work that we do here, which you mentioned at the end, should we actually be saying climate change and ocean acidification as being two elements of the problem caused in different ways by CO2 emissions? Thank you.
Thank you very much for the question. No, I think I use the two terms just to identify ocean acidification as a subset of climate change. So with climate change, I just want to highlight all the possible changes that are linked with the increased carbon dioxide in the atmosphere. So the warming, the acidification, the changes of the carbonate system, and other things that we may not take in consideration. Whereas ocean acidification is a specific refer to the pH, the increase in the pH. And with regards to the metals, because they are present in different chemical forms in the water, of course they are more affected by the changes in the pH because they alter the ionic form. But at the same time, the complexity of the climate change will affect the health and the capacity of the invertebrates to cope with this toxicity. So it's really a combined effect of the two. Thank you.
Thank you. Any other? Sorry. Germany?
Thank you. Thanks, Andrea, for this very impressive work and presentation. I have a quick question. What does it tell us about proactive management of contamination? We mainly do this in Europe currently using environmental quality standards. I know that we take bioeffects in addition, but they have some challenges because they integrate signals and you need, as you've shown, a careful analysis to then take it apart and see what pressures there are. From your work, I would think that we would really need to lower the levels of environmental quality standards for instance, for the metals, to reduce the exposure of the organisms, to be proactive. Would we have a clear indication from such work by how much? Because it's also like a huge political issue, but what is your take on this? Thank you.
Thank you very much for the question. Not so easy to answer that. My opinion is that, yes, that would be the correct approach, but realistically, we still don't know. Because apart from the interaction between climate change and the the chemical interaction of climate change and the contaminants, then there will be the biological stress of climate change and the contaminants, and then there will be the interaction between the contaminants themselves. So to understand how to put all this information together to pass from one value X to value Y will be very challenging. The call is really we need to consider these aspects and we need to work on these values. We can't leave them as they are, that's for sure. There will be, all the biodiversity will be more sensible. That's the key final message. I can't estimate how much, but you know, that's what we have to discover and that's what we have to work for, really. Thank you.
Thank you. Oh, yeah, EDF.
Thank you for this talk. That was really interesting. I just had a question about, you know, what life history forms of these different species that you were looking at with ecotoxicology. Some species and then some, you know, juveniles or larval stages can be more sensitive. Did you look at those as well? Thank you.
We cover the larval stages in the first work that we've done. So that's a standardized ecotoxicological test where we collect the gametes from the sea urchin and we expose the larvae because they're very sensitive, as you said, to contaminants. In this second experiment, we just use adults of mussels. So because they were in the mussel farm and we wanted to standardize the sampling, we just use the commercial size, which is roughly, I think, 70% of the maximum length of the mussels, between 3.5 and 4.5 centimeters, if I remember correctly. But they were adults. So in the first study, we used the larvae, and the second one, the adults. Of course, all the larvae should be tested because, as you said, they are very, very sensitive. So. Yeah, but the results on the iron sulfate toxicity, that as well was run on larvae of sea urchin.
Thank you. I think we have to move on. So if you have more questions, maybe you can ask him in the discussion time because he's here with us today. Okay, thank you again, Dr. Guyon. Okay, now I would like to introduce our next online speaker, Dr. Andreas Oscholitz. Dr. Oscholitz is a professor of marine biogeochemical modeling at GeoMar, Helmholtz Center for Ocean Research, Kiel, and the University of Kiel, Germany. His research examines global carbon, nitrogen, and oxygen cycles, their sensitivities to environmental changes, and the development of numerical models to assess these processes. He chaired the Collaborative Research Center on Ocean Deoxygenation and initiated the Kiel Declaration on Ocean Deoxygenation. He is co-chair of the Global Ocean Oxygen Network and the Global Ocean Oxygen Decade Program, and co-chair of the German Marine Research Alliance mission on marine carbon sinks. The title of his talk is Potential Impact of Marine Carbon Dioxide Removal on Ocean Oxygen. Professor Oscholitz, you can start when you are ready. Professor, you are muted. We cannot hear your voice. We still cannot hear your voice.
Okay.
Oh, yeah,
Thank
yes,
you
we
very
can.
much for unmuting me. Sorry, I couldn't do anything. I share my slides again. Apologies for these technical issues. Okay, now you should be, I hope you can see my first slide and you can hear me. And otherwise, interrupt me. I will just continue then and start my presentation.
Yeah.
This is a joint work of the Global Ocean Oxygen Network, GON, and the looking at oxygen changes on the ocean, and the CDR-MARE community, a German research program on carbon dioxide removal. And most of the results I will show here are already recently published in a paper where the QR code is on the bottom right. And so this is all about oxygen, another dimension of current ongoing anthropogenic climate change. We have warming, we have acidification, but the ocean is also losing oxygen at accelerating speed. And this is the from the last IPCC report, the global inventory of ocean oxygen. So it is declining by, we think, several percent over the last decades. So it's still a substantial amount, maybe not lethal right now, but there are some risks that we may run into losing substantial parts of habitats of marine life due to oxygen loss. So if you cannot breathe, nothing else matters. And this is really a threat for large parts of ocean ecosystems. The main cause of this is global warming. That's what we see from climate models. That's the current best understanding we have. And on this slide, you see the different curves of the IPCC scenarios, higher emissions, redder curves, higher temperatures, but also lower oxygen loss, larger loss of oxygen. So stronger warming induces a larger loss of oxygen into the future as well. One problem we identified is that current observational estimates show a much faster decline of oxygen than current climate models do. So there's still a factor 2 to 3 discrepancy, and this is also alarming and is telling us something about the confidence we can have into current projections. Oxygen is closely related to temperature and to also the carbon cycle, which we seem to get right on the global average, but these oxygen measurements, which are pretty precise, and having been done with the same approach for more than 100 years now, the Winkler titration method, show us that oxygen in the ocean is declining faster than our best models today project and simulate. And we seem to have some underestimation problem, but still we need to understand what are we doing, and we have to mitigate deoxygenation, oxygen loss, which has in the geological past of the planet led to some major mass extinction events. We have to find ways to stop this as quickly as possible. And because it's caused by warming, plus in some coastal areas, eutrophication, so nutrient input, we also know that we have to stop warming or reduce warming as quickly and as possible. So reduced warming will lead to reduced ocean deoxygenation. And this is where now carbon dioxide removal may play some role as well, in addition to, of course, emission reductions, which is the first and most urgent activity we have to start and engage in. Has to be remove 90% of our emissions, but the remaining residual emissions, there if we want to meet climate targets and also stop oxygen loss in the ocean, we have to apply carbon dioxide removal according to current knowledge. And here's a sketch of different methods that have been proposed. And in our study, we now looked at not only what is the mitigation potential in terms of carbon dioxide removal and reduction or mitigation of additional warming, but also what is the mitigation potential in terms of oxygen loss. And some of these methods do have, of course, side effects in the ocean. They also lead to additional oxygen loss or a different carbon or chemistry in the ocean. And this is the aspect of the study. We looked mostly at, or only at open ocean methods. There are coastal blue carbon methods, mangroves, salt marshes, seagrass meadows. They also have oxygen issues. They produce oxygen during the day when they do photosynthesis. It's biology. Photosynthesis produces oxygen. But at night, they respire. When there's no light, no photosynthesis, oxygen will be consumed by this newly produced, during the day produced fresh organic matter. And so these coastal systems, blue carbon systems, tend to amplify the diel cycle, so more oxygen during the day, which also at high concentrations can become toxic, but also less oxygen at night because of the additional production of carbonic molecules that are then respired in the dark at night. So this is also some aspects we have to consider. But now let's look at the open ocean approaches. Iron fertilization has just been mentioned in the previous talks. Is one method where the Conventional London Protocol has already achieved a relevant, significant governance scheme. There have been a couple of 13 field trials, all mostly in the Southern Ocean, but I'd highlighted this in the Arctic as well. And they have shown there are major ecological shifts, of course, because that's intended to fertilize the ocean, to make biology more productive and hopefully draw down carbon. There are many questions about durability, additionality, and so on. And here we look in now model studies, these what-if scenarios. What would happen if we applied ocean iron fertilization in the Southern Ocean, where it has the largest leverage in terms of carbon dioxide removal? What would happen in this ocean to oxygen? And the scale here in all these model simulations we do is about 1 gigaton carbon drawdown per year. So it has some significant impact and climate mitigation potential. And we see in these maps, the lower right panel is a global map, what the vertically integrated or averaged oxygen change in the ocean after 100 years of iron fertilization. So a very hypothetical experiment, but still at a significant scale. That is also mentioned in many of these reports. We need gigatons per year carbon removal. And that would be the result in terms of oxygen. So we lose oxygen a couple of 10 millimoles per cubic meter or micromole per liter. The upper right panel shows the zonal average, and we see mostly in the Southern Ocean. On the left, where iron fertilization is applied, we have a drawdown of oxygen by some up to 50 micromole. It's still not going anoxic, but it's a systematic decline in oxygen loss. And it will affect ecosystems. They live also, some species live at the border of existence. They have high competition. They cannot swim as fast. They cannot breathe as much. So they will feel this change. On the global average, what happens to the inventory of the ocean in terms of oxygen content? The black and blue lines show the decline under ongoing business as usual scenarios. So we see this continuous decline, what the IPCC models, CMIP models also project. And the green is the decline if we also apply our ocean iron fertilization. So we see an acceleration of the oxygen loss in the global ocean, which is outcompeting the slight gain of oxygen we would have if we had reduced emissions by the same amount. So basically achieving the same climate, not by ocean iron fertilization and its projected CO2 drawdown, but by reducing the emissions by an equivalent amount. This is about 1 petagrams of carbon per year. And this is the dashed blue line. So we see a slight gain or a slightly less strong decline in oxygen if we have a reduced warming. That's what we expect from the IPCC models as well. Less warming means less deoxygenation. And that's the slight discrepancy here between the black and dashed blue line. So a slight gain in oxygen compared to the control, to the baseline. But if we apply ocean iron fertilization, because of all the respiration of the newly produced organic matter in the deep ocean, we have a drawdown of oxygen. So it's basically a eutrophication, intended eutrophication by ocean aerosolization. And this is about, well, it's a rough, drastic acceleration of deoxygenation. Another method that is being studied right now has been proposed is macroalgae farming, and it also has, well, photosynthesis in the surface ocean where we have light, nutrients, and water there. But this organic matter, if it is sunk to the deep ocean where it will eventually respire, would also consume oxygen there. And this is again shown in the same kind of model simulations here, about on this 100-year time scale and 1 petagram per year intensity scale. It's basically global activity, hypothetical. We see a loss of oxygen in the map almost everywhere in the global ocean because these macroalgae can grow in many places, not only the Southern Ocean. We seed them here wherever there's enough nutrients in the surface waters where they can grow and have annual net production. And we see also oxygen decline in the upper right panel, mostly in the deep ocean where these algae are then sunk to and where they respire eventually. There's a slight oxygen gain here in the upper ocean because these macroalgae just sink through very quickly, and so they don't respire in the near surface waters, but they respire at a deeper depth level. So that shifts the oxygen loss mostly to the deep ocean, where we also will hit the deep ocean ecosystems, but if we applied this macroalgae sinking and respiration. And again, the same figure here. We have the baseline run, the black curve, business as usual, oxygen decline. And macroalgae was where we assume different sinking, different remineralization patterns, either the entire biomass or just the non-cellulose part. That's the orange curve, but we see a net acceleration of oxygen loss, particularly in the deep ocean. What happens if we do not sink the macroalgae but harvest them and use them for bioenergy on land and plus CCS if available at these scales? Then the picture is very different. Then basically we do not allow respiration of the additionally produced biomass in the ocean, but we have the burning of these biomass on land and controlled facilities, and ideally in these scenarios with CCS, so no CO2 escapes to the atmosphere, no additional warming from that. And thereby we take out, we avoid respiration in the ocean, and so we avoid oxygen loss in the ocean. And in this scenario where macroalgae grow in the ocean, are harvested and used on land for CCU plus CCS, that is carbon utilization and storage, then we have net gain of oxygen for the first time. So this is one method where we could really almost stop deoxygenation. That's this yellow curve here on top, where respiration in the ocean is avoided, therefore oxygen loss is avoided, and the warming is slightly reduced as well on this petagram carbon per year scale, carbon dioxide removal. So this is one method that might work from an oxygen perspective. Of course, we would take out nutrients from the ocean and many things would change as well. Ecosystems are affected, will change, biodiversity and so on. But oxygen-wise, this could be a game changer and could really stop deoxygenation. And now all the results can be combined in one diagram that looks at first sight complicated, but I will talk you through. On the horizontal axis, we have the reduction in atmospheric carbon, atmospheric CO2, in these applications. We see 100 petagrams, 200 petagrams, so it's about 1 petagram per year carbon reduction removal from the atmosphere. This would be about a substantial part, 10-20% of our residual emissions that we think we have to compensate by carbon dioxide removal. On the vertical axis, we have the oxygen, net oxygen change in the ocean's oxygen inventory. So this is an oxygen loss, the red part, and the blue part to the top would be an oxygen gain. Where we start is the black lines, that's the historical change. So we have increased atmospheric CO2, we go to the right on the axis, and we have decreased, we go down and the vertical axis, ocean oxygen. This is what we have already done to our oceans and to the climate, injecting CO2 into the atmosphere, reducing oxygen. What happens now for the carbon dioxide removal? First, ocean iron fertilization and macroalgae sinking. So these biotic methods, producing more biomass in the surface ocean, sinking it to depth where it will respire. It does. These curves go from today's point on the right part of this figure here to the bottom left. So they reduce atmospheric CO2. In the model, at least, they can work. But they also reduce oxygen by about 30 to 40 times faster than current deoxygenation that is going on. So it's a substantial, steep slope here in oxygen loss that we have to take into account. If these methods work, if they remove carbon from the atmosphere, and if the biomass is respired aerobically, then this will lead to a major loss of oxygen. If only the soft part of the algae is remineralized, then this slope is still substantial. We still accelerate the oxygenation, but less severe. The blue curves here are ocean alkalinity enhancement. These are geochemical methods. They do not rely on photosynthesis, not on respiration. They just change the chemistry of the ocean. That was at the end of the first talk, I think, the compensation of acidification by dissolving limestone, for example, in the ocean. That is ocean alkalinity enhancement. It does not directly impact oxygen. It slightly increases oxygen because of the reduced warming. So that's compared to the baseline, one way to slightly remove the oxygen stress or mitigate the oxygen stress in the ocean. And the last part is this macroalgae harvesting, so taking out biomass from the ocean, avoiding respiration of the ocean. That could lead to a net gain of oxygen in the ocean. So this calls for a consideration of oxygen, really, for all these carbon dioxide removal methods that at least target the ocean. If we apply a different metric, only suboxic volumes where fish cannot breathe, so low oxygen levels, in our case here, it's 5 micromole for those interested, and we have the area in different depth levels, so that's the vertical axis is depth, and the percentage of the current ocean surface, these are the black and blue lines here, so it's about 2% in certain depth levels, which is not a habitat for fish because too low oxygen. And we see here many methods like ocean iron fertilization and macroalgae. They reduce the extent of these suboxic volumes because they shift the respiration to deeper depths generally. So they release pressure in these upper 500 or several hundred meters near surface waters where we have currently oxygen minimum zones. And so there could be even some mitigation of oxygen stress in these near surface waters on the expense of a larger oxygen loss at depth. Some methods like artificial upwelling, I haven't mentioned that earlier, they would completely create havoc for oxygen minimum zones and expand those quite a bit, but that's not on the agenda anymore, I think, in most discussions anyway. So the summary here, we see that biotic carbon dioxide removal methods, iron fertilization, macroalgae, but also blue carbon methods, they do change oxygen. That's intended. They produce oxygen during photosynthesis and remove oxygen during respiration. So we have to consider that. And marine oxygen loss generated by these methods can be many times larger, so 30, 40 times larger than the small oxygen gain because of the small reduction in global warming of these methods. Abiotic methods like ocean alkalinity enhancement or electrochemical methods, they do not directly target photosynthesis, they do not target directly biomass, and therefore not respiration, and therefore not oxygen. So they may be less problematic from an oxygen perspective. Macroalgae farming and harvesting, so taking out biomass from the ocean, might be, from an oxygen perspective, a mitigation approach to mitigate deoxygenation. Of course, it would also take out ecosystems, nutrients, and change the ocean in many other ways. And certainly at the end, oxygen should be one of the key variables in terms of monitoring. It's easy to measure. We have a very good baseline, long historical records. It's a cheap measurement, and we just need more data to assess our ecosystem changes and the habitats in the ocean by having more information about oxygen. And that's, if you would like to have more information, you can read a little more here in this paper. I thank you for your attention, and again, apologize for the disrupting your schedule, which was very well planned, of course. Thanks for your attention.
Thank you. Thank you very much, Professor Schuris, for very interesting presentation. Now we have time for questions. Italy?
Thank you very much for the very interesting presentation. I have a question with regards to what may be the implications of altering the oxygen levels with CDR techniques in a quite fast manner with regards to the redox potential of the seawater, because we know that altering the redox potential can alter the toxicity of metals, for example. So I'm just wondering, when we change the oxygen levels, if that can affect redox and toxicity as a consequence? Thank you very much.
Yes, thank you. Yes, of course, it will affect all other redox-sensitive processes, and toxicity was certainly one major aspect. It also can or will change the availability of nutrients. So there's the sediment interface, iron release, phosphate release is oxygen sensitive. So there are a couple of additional changes down the pipeline, feedbacks in the system via redox changes. In general, that's what we see right now. So there are likely some positive feedbacks to ongoing ocean deoxygenation via more nutrient release from the sediments, turning anoxic. So that might be one explanation why current models underestimate the observed decline in oxygen. So there are very good reasons to mitigate deoxygenation. And if carbon dioxide removal methods can mitigate deoxygenation, this might be an added value we have to take into account in our assessments, I think. So it's more turning back towards pre-industrial. I wouldn't say we need more oxygen in pre-industrial oceans. So it's really a mitigation, on more fronts than just carbon dioxide and temperature.
Thank you. Yeah, ocean care.
Thank you, Dr. Oschlies, for another excellent talk of yours. I would agree that oxygen is a key variable that we should be measuring concerning ecosystem change. I would like to add a couple of nuances to the descriptor of ocean alkalinity enhancement, which I think might come across in a rather favorable light here. I mean, I think one comment I would make is that it's unclear if we could actually determine what quantity of sequestration would ever be done by doing OAE, but that's not really the focus of my comment today. You said it's a less problematic technique in this regard, and I think that's fair, but the assumption that OAE would primarily be a geochemical intervention is correct, but it is possible that subsequent ecosystem responses could arise doing OAE that could alter primary production, export fluxes, respiration, which would then have deoxygenation impacts. And if silicate materials were used, the release of nutrients and trace metals could also stimulate biological productivity and therefore impact oxygen consumption. And as I understand, the assessment focuses on global averages, which may obscure some of the regional considerations here. So if we were to do OAE, for example, in coastal zones or upwelling systems, relatively small changes in productivity could potentially exacerbate local hypoxia, which we see already in those systems. Thank you very much. I'd appreciate your thoughts. Thank you.
Yeah, absolutely correct. So I didn't want to advocate for anything here, but it's. I think for OAE at least, it is not intended to modify, to change ecosystems and to change oxygen levels. Whereas for a biotic method, it is intended, biotic marine CDR methods intend to modify ecosystems to enhance production and thereby to enhance respiration and oxygen consumption, if they work. So that if they work, they will lead to less oxygen. For OAE, it is not intended. It doesn't mean it doesn't happen, but there's at least some hope it can be done, and it could be done in some responsible way where oxygen changes can be minimized or can be reduced to some amounts deemed acceptable. It is, we have to assess them in many fronts, but there we do not automatically reduce oxygen as well.
Germany?
Thank you. Thank you, Andreas, for giving this presentation and for your time and willingness to show this very important work here. I have a quick question with relation to, I think you made a really good point, how important oxygen is. And in this increasing discussion of which methods should go forward of these MCDR approaches from moving to research, from research to deployment, I think we also started a discussion on showstoppers. So for my understanding, oxygen could be a potential candidate. And having said this, of course, there's some nuances to this. But my question related to this is, putting it very bluntly, we've been as humans very successful to deoxygenate our ocean. So we have quite large naturally oxygen-free areas, dead oceans, but also anthropogenically oxygen-free areas. And we have the deep sea. And there, the respiratory processes are either stalled, or oxygen is already depleted, or they're even slower. And if we only focus on these regions, did you, in your study, take this somehow into account? Because these areas are quite large. So how would this factor into the equation? Thank you.
Yes, we looked at this. So it's oxygen. Well, the deep ocean right now, apart from basins like the Black Sea, is pretty well oxygenated, and it stays so. In all of these simulations, we don't find additional anoxic areas in the deep sea. So it's not. We don't see the development of sometimes called dead zones there. But we see additional stress on ecosystems. So a reduction by 50 micromole would impose substantial stress to the ecosystems and very likely change the composition of ecosystems. It also, may lead to smaller body sizes, and there are several theories out there. I think it's, we have to account for it. Climate-wise, it would be good to have anoxic oceans. Then a lot of carbon can be stored there. So that's how the Earth system in the past has mitigated climate catastrophes, by running into anoxic oceans and storing all the carbon, eventually leading to fossil fuels by accumulating unrespired organic carbon in the deep sea. The question: what does society want? And it's a responsibility issue. We have winners and losers all the time. We shift ecosystems, and I think therefore these geochemical methods, in my thinking, can be probably be applied in a more responsible way, because they do not intentionally change ecosystems. They may still, we have to be very careful there, change ecosystems, but there is some hope that we do not intentionally modify ecosystems, which might in many places go wrong, and ecosystems are difficult to predict, more difficult to predict than chemistry. So there are some qualitative changes, so that these green methods that we like, or society often likes, because it's what we do in our gardens, may have at these large scales required for the petagram scale or gigaton scale, may create substantial changes in other parts of the ocean. The deep ocean is something, deep ecosystem you ask about is what we really know least. Where we even don't know the oxygen thresholds of most of the species there. We have some information about near-surface species, fish, what are their oxygen tolerance levels, but for deep ecosystems, we have very little information, and it's therefore, we should be very cautious to make strong inferences about deep ocean changes, which we understand not very well.
I see no more questions. So thank you again, Professor Oshiris, for very interesting and important presentation. Thank you.
Thank you. My apologies again.
Now we are a little bit behind of our schedule, but I think we can still have time for coffee break for 20 minutes. So maybe we can resume. We will resume at 11:30. Thank you.
haven't got time now anyway.
Hello. Is there Professor Lucas in the meeting room by any chance? Oh no. I think it's not. Hello, is Dr. Lucas Pose in the meeting room? He's not answering.
Okay, welcome back. Because most of our speakers are participating online, so it may be difficult for them to remain available for the general discussion session at the end of the program. Therefore, instead of reducing the Q&A time following each individual presentation, we may reduce the time of the discussion session. Okay? And so now our next speaker is an online presentation by Dr. Lucas Porch. Dr. Porch is a postdoctoral scientist at Helmholtz Zentrum Hereon, Germany. His research investigates how human activities alter seafloor habitats, sediment transport, and the marine carbon cycle, with emphasis on the North and Baltic Seas. He is actively involved in several expert groups of the International Council for the Exploration of the Sea, contributing scientific advice to policymakers on sustainable marine management. The title of his talk is Dredging and Dumping Impact Coastal Fluxes of Sediment and Organic Carbon. Dr. Porch, you can start your talk when you are ready.
Yes. Hello, everyone. Can you hear me?
Yes, we can hear you very clear.
Okay, I still have to share my screen, I think.
Yes.
Okay, yes, thanks for the introduction. And I'm happy to share the results of a study we recently did on this topic. So you can see the list of co-authors that contributed below. And before I get into it, I just some definitions. So I'm talking about dredging and dumping. And so what do I mean? I'm talking today about two separate activities. So the first is the dredging in form of material extraction. So that means mainly sand and gravel mining for use in harbor construction or beach nourishment. So this is the first thing. And when I talk about dumping, I mean the disposal of material taken from or during waterway or harbor maintenance. And so when I say dredging, I mean the first thing. When I say dumping, I mean the second part. Okay, so now that's clear, hopefully. So the motivation behind this study was actually related to blue carbon. So blue carbon was explained earlier, I think, refers to the capture or the uptake of atmospheric CO2 by coastal and marine habitats and sequestration in the long term. And this sequestration takes place mostly in the sediment. So the carbon is stored in the seafloor. And that's why we are mostly interested in the seafloor. And of course, we know that humans relocate a large amount of sediment, but it's not really clear how much sediment that is, and is it really important in terms of carbon fluxes? So we wanted to know how large are the impacts, especially compared to the natural fluxes, or do we need to worry about them, or can we ignore them? And finally, how much of the disturbed organic carbon is remineralized to CO2. So that means what is the ultimate climate impact that we can expect from these activities. Okay, so our study area was the North Sea. So the North Sea, you can see a map here, and it's of course very heavily influenced by human activities. And at the same time, we have a lot of data. So both on the natural dynamics are quite well understood, but also we have a lot of data on the human activities. So what you can see here, the background color that you see here, this red and orange, indicates the vulnerability of the sedimentary organic carbon. So that means in those red areas or dark red areas, when the seafloor is disturbed, there's a high likelihood that it will release a lot of CO2. Whereas in the lighter areas, so down here, these are rather sandy areas. If the sediment is disturbed there, there's very little carbon or it's not so reactive. So disturbance there will not have such a large impact on remineralization. And this is mainly important for aggregate extraction. So you can see here the blue polygons. These are the aggregate extraction areas in the North Sea. So this is where sand and gravel mining are permitted. And the green dots here are the dumping locations, or they're not dumping locations, but I have aggregated them here spatially so they don't overlap too much. And so you can see that everywhere where we have large harbors, of course, there's some dumping activity nearby, mostly at the Dutch and Belgian coast, but also quite a bit of activity at the German and UK coast. And most of this data was taken from OSPAR and ICES reports. And this carbon reactivity map was a result of an earlier study that we did. Okay, so how do we quantify these effects? So we use a numerical model. So that's a process-based 3D hydrodynamic model that is coupled to a sediment transport model and also a benthos model, so simulating the ecosystem functions of bioturbation. And so on the left here, you see the total or the entire model domain, and it's encompassing the Northwest European shelf and also part of the North Atlantic. And on the right, you see it's still depth shown here, but it shows also the grid. So I don't know if you can see it clearly, but there are all these triangles. And on the shelf, they get more dense, so the resolution is increasing. And then we have a special area of interest here in the German Bight, including the Wadden Sea, so the system of tidal flats where the resolution is increased even more, and that's horizontal resolution is a few hundred meters. And yeah, this model, the hydrodynamics and sediment dynamics, have been extensively validated in previous studies. So we are quite confident that it's able to represent the natural dynamics of the system. Okay, so how do we get now the dredging and dumping impacts inside? I tried to sketch it here. And so on the left, we see the sand extraction process. So this means a removal of the sand and gravel fraction from the seabed, and simultaneously the fine grain fraction, so this includes mud and also the organic carbon, is re-exposed to oxygenated bottom waters, so the remineralization rate can increase, and the carbon is ultimately or can be lost as to CO2. And when there are currents, it can also be transported and redeposited elsewhere. And then, so this is for sand extraction. For dumping or disposal, yeah, it's very straightforward. It's just an additional point source of sediment and organic carbon at the sea surface, which is then sinking down to the seafloor and can be, in the absence of currents, can stay there or be resuspended and transported and redeposited. Okay, so this is how we implemented it. And now I come to some results. So first we look at the total amount. So you see here a time series starting in 92 to 2023. This is for material extraction, and we can see a slight increasing trend here over time. And we also see some years where the amount really more than doubles compared to the long-term average. And this can be explained by some very large projects which were done during those years, like the construction of a port or some large sand nourishment projects. And so the left y-axis here shows the total amount of extraction, so the bulk sediment, And the right axis shows our estimate for the organic carbon in this extracted material. And you can see that, first of all, it's few orders of magnitude lower, since in sand and gravel there's not much carbon usually contained. And then we have these different colors, and they correspond to different reactivity classes. So according to this map that I showed earlier, so how much of it is very reactive or not reactive. And you can see that most of this disturbed carbon seems to be in the lowest reactivity class. So this is already a first indication that maybe this extraction process does not cause too much trouble in terms of carbon. And in fact, this is confirmed by our simulations. So here you see the result after 1 year of continuous extraction. So you see some areas which are red. So this is where we have a loss of sedimentary carbon. And the blue areas indicate a gain. So the reason there is a gain in some areas is because of this resuspension and transport and redeposition. But you can see that this color mapping is also logarithmic. So these blue areas are really only a very minor amount. And in the end, or at the end of this 1 year simulation, we have around 1,000 tons carbon removed from the sediment, compared to the reference state without any extraction. And yeah, we see this slight relocation. But yeah, this 1,000 tons is, I would say, quite minor amount. So yeah, this does not seem to be a huge issue, even though the amounts of bulk sediment, of course, are very large, which are relocated here. And so with that, I come to maybe the more interesting part for today, which is the impacts of dumping. And here the data is a bit messier, so I will take some time to explain. So here again, we see a time series, this time 95 to 2021. We see some years where this OSPA data is missing. And we also see that in 2008, the numbers seem to go up, but this can be explained by the fact that the UK decided to supply data starting from this year. So this explains most of this increase. And yeah, what was surprising to us is that we don't see a very strong increase in, or in fact, even a decrease in the last 10 years or so, which was surprising because we always hear about extension of harbors and deepening of waterways and so on. So we would expect this to go along with higher amounts of dumping, but at least in this time series we cannot see it yet. And then in 2013 we also get some information on the sediment composition of this dumped material. So whether it is rather coarse or sandy or gravelly, or whether it is fine, so mud. And this is important for us because it allows us to better estimate the amount of organic carbon contained in this dumped sediment. So muds tend to have a higher organic carbon content compared to sands. And so this is what the black line indicates. So this is the organic carbon in the dumped material, and the uncertainty is shown here by the shaded area, so it's still quite high because we just don't have that much measurement data on organic carbon in harbor muds or in dredged harbor muds further. And if we look at that spatially, so here it's zoomed in onto the southeastern part of the North Sea, which is our area of interest, and which is also a dumping hotspot, and where our model is highly resolved. And so some of the large ports here are labeled, and you can see that a lot of the dumping, especially in the Wadden Sea area, so in the German Bight, is really going on inshore, so in the estuaries or very close to the coast. Whereas farther south towards Netherlands and Belgium, there's also quite a bit of dumping offshore in the open sea. Okay, so here I show the dumping model results. So here's panels, and these show the distribution of dumped material, of fine-grained dumped material after 1 year of simulation. And because the dynamic behavior of this dumped material is not very well known, we did a series of experiments varying the sediment sinking velocity and where we inject the material, whether it's at the surface or the bottom. And I show here 3 instances, and you can see that the patterns really change between them. So it really, it's quite sensitive. However, what does not change so much is how much of this dumped material ends up in the nearshore. So in each of these, the amount depositing in the Waddensee, so this is this Waddensee World Heritage Site, which is outlined in blue shading here on these maps. And I should also say the blue dots are the individual dumping locations that we have defined in the model according to the data. And so around 60 to 70% of this material, which is dumped in this Waddensee area or around that area, finally ends up in the Waddensee. And this happens to be a similar amount as has been estimated for the total mud budget, so the total mud influx into the Waddensee annually. So this already gives us some indication that that this human activity is really as important as the natural fluxes. And what is also notable about this is that a considerable part of this dumped material ends up in the intertidal zone, so actually on the tidal flats. And we know that the tidal flats are accreting a little bit along with sea level rise. And so it's an indication that already now this dumping activity can be somehow supporting this accretion of the tidal flats. Okay, now we want to compare this to other fluxes and impacts in the North Sea, so both other anthropogenic activities as well as some natural fluxes, and I list them here. And I should say that most of these numbers also come with high uncertainty, so I think we should just look at the exponents here. So this is in kilotons bulk sediment per year, and in the top rows we have some human impacts. So starting from bottom contacting fisheries, so bottom trawling basically, that is around 1 million kilotons of bulk sediment disturbed in the North Sea each year. Dumping impact is not quite as high. Aggregate extraction also similar. For offshore wind farm construction and telecommunication cables, we don't have reliable data, but I would assume that it's much smaller than these other ones. And when we compare them to the natural fluxes, we see that they are at least of a similar magnitude as, so the human impacts are of similar magnitude as the natural fluxes. So this includes coastal erosion, aeolian deposition. So this means atmospheric dust deposition, basically. And also river export, which is normally seen as the main kind of conduit for the main source of sediment to the sea, as well as marine inflow and marine outflow. So this is the flux to and from the Atlantic, and also sedimentation. So all of these are kind of similar in magnitude, but many of them smaller than the human disturbances. We can do the same for organic carbon flux, and here the numbers are not so impressive, let's say, but still in some cases similar to the natural fluxes. But here the natural flux is really dominated by the marine in and outflow from the Atlantic because of the stuff that is in suspension, and this is just a huge amount. So we see that dumping and bottom contacting fisheries are highest on the list, and aggregate extraction is an order of magnitude lower at least, and these construction activities are even smaller. Okay, so looking globally, of course, as we look globally, the data situation is not really improving, but so the uncertainties are very high. But there's one data set, at least from Marine Sandwatch. So this is a UN Environment Program project which tries to aggregate all of the marine sand mining activities. And so you can see that we have it here on a global map, and we can see the highest sand extraction is going on in the North Sea, but also a lot in North America and China and some other areas. And if we just try to upscale our results from the North Sea globally, we find that we get a range of around 0.09 to 0.46 gigatons carbon per year, which is disturbed here. And again, we can compare it to the global river organic carbon flux, which has been estimated to 0.42. So another indication that the dredging and dumping plays a major role here on these coastal carbon fluxes. Okay, so to summarize, we found that coastal sediment redistribution by dredging and dumping may be as important, or in some cases even more important than the natural processes. We estimate that dredging and dumping have a larger carbon impact than marine construction, but smaller than bottom contacting fisheries. We found a small impact of material extraction on sedimentary carbon, so this around 1 kiloton per year in the North Sea, just because it's mostly sandy. And we found a large disturbance of sedimentary carbon by dumping, but we chose not to define the impact on the carbon cycle here because a natural baseline for this dumping activity is really lacking, because the harbors themselves are, of course, not natural systems, so it's really difficult to define a reference state here. And for an outlook, the One Sea World Heritage Site is, of course, supposed to be somehow protected, but in practice, we saw that even today, a lot of the dumped material seems to end up there, according to our model. So the question is, what are the ecosystem impacts of this? Possible ones could be the smothering of benthos, local habitat alteration, so going from sandy to more muddier sites. And the increase in SPM could also cause some oxygen depletion at the bottom. It also raises the question whether these human impacts should be considered in blue carbon evaluation schemes, so both in the nationally determined contributions, and there are also some ideas to do a monetary evaluation of this blue carbon. But I think this becomes very difficult if we consider also these human impacts, which might be more important than any sequestration by seagrass meadow or something like this. So I think this could be an important point. And it also could mean that this dredged sediment could be of beneficial use to help the growth of tidal flats as they struggle to keep pace with sea level rise. So this is something we are currently looking into, if this is feasible. And if you're interested, I invite you to take a look at the paper. And otherwise, I hope there's time for questions, and I thank you for listening.
Thank you very much, Dr. Lucas, for very impressive presentation. We have time for a couple of questions. Yes, UK.
Yes, thank you, Chair, and thank you to the presenter for a really interesting presentation, and incredibly useful as well. Some really interesting insights on sort of dredging and dumping as in better understanding dredging and dumping from the sort of historic, fairly simplistic sense of a purely polluting activity to quite a major process that can exert all of these different kinds of changes, which could be positive, could be negative, and depending on how you depending on the matter at hand. I had a specific question on, I guess, the parameters going into the modelling. were you able to differentiate between dredging method and then dumping method? Noting that obviously certain hydraulic methods will have much greater oxygenation, because they're so much more disturbing to the actual sediment habitat compared to mechanical methods. And whether you were able to look at different dumping methods, particularly when you looked at different modeling scenarios with different injection sites, because obviously there are data out there as to how much of the dumping we do is by bottom dumping versus so-called rainbowing, etc. So I just wondered whether you were able to consider that in your modeling. Thank you.
Okay, Dr. Lucas, when you are ready.
Okay. Yes, great question. Thank you. So the short answer is no, we did not consider these different types of activities. I should note that in our model, the very near shore, where maybe rainbowing is more prevalent, is not very well resolved, and so we just put everything at the sea surface. And in terms of dredging, I think the most prevalent type of aggregate extraction is basically this trailing suction hopper dredges, which disturb around the top 30 centimeters. And this is what we implemented in the model. But I think this is a good idea to look at this in future.
Okay, thank you. I see no further flags. So thank you again, Dr. Lucas, for very good presentation.
I think there's one online question.
I've got a question for you, Lucas.
We have a question from Dr. Hal Spencer. Okay.
Yes, thank you. I noticed there was pushback from the fishing industry about the amount of carbon released by bottom trawl activities. And the argument seems to hinge on whether that carbon is available for breakdown by microbes and other organisms, or whether it's refractory. Did you look at that aspect, please?
Yes, thanks for the question. Yeah, exactly. So actually, these numbers that I showed for bottom trawling were from our own study, so not from the original one that had these huge numbers, but from our own one where we did consider this lability problem of the sedimentary carbon, and indeed the numbers were much lower than the original ones, but still higher than what we saw now from dredging and dumping. So we did consider this, and there's still, of course, uncertainty, but even these refined numbers seem to still be considerable. And also of course, the dredging area, or the trawling area is much, much larger than anything that is affected by dredging and dumping. So locally, the impacts will be, of course, very large. Thank you.
Thank you again. Now we have to move to next speaker. Our next speaker is Dr. Douglas Wallace. Dr. Wallace is Canada Excellence Research Chair Laureate and Canada Research Chair in Ocean Science and Technology at Dalhousie University. He also serves as Scientific Director of the Marine Environmental Observation, Prediction, and Response Network. He has authored more than 130 peer-reviewed publications. He previously served as Professor of Marine Chemistry at GeoMar. The title of his talk is Ocean Reoxygenation. Dr. Wallace, you can start when you are ready.
Now I'm unmuted. Thank you. So, yes, good morning, everybody, and thanks to the organizers for inviting me, and especially for organizing such an interesting session. I'm joining you from Halifax on the Atlantic coast of Canada, or the east coast of Canada, so the session started a little bit early for me, but thanks to the previous presenters for their interesting talks, which both woke me up and kept me awake. And thanks especially to Andreas Oschlies for setting the scene with respect to the threat and the issues related to oceanic oxygen. I'd like to acknowledge my co-author on this presentation, Patricia Handman, and I'd also like to, if possible, add a question mark, really, to the title I gave earlier. And the question mark is put there because this issue is something that we've actually not really yet attempted in the ocean, at least on any significant scale. We've barely studied it. And yet I think we believe it's an important issue to consider and timely, maybe even urgent to consider. So thanks for the opportunity to talk about it. I realize I should have started my time. Okay, so I think we're all, pretty aware of the chronic changes to the Earth system arising from global human impacts, and that inexorable increase in temperature, in acidity, and also in decrease in oxygen, which Andreas talked about, all of which are major threats to the ocean and its ecosystems. But arguably, it's really the extreme events, their increased incidence and their increased amplitude, which is of major concern for marine ecosystems, including synergistic effects, which were mentioned in a couple of the earlier talks. And this paper, published 2 years ago, had a big impact on me, really, in talking about these extreme events impacting the ocean, and pointing out how they're substantial risk to the environment, and recommending 3 kind of threads of research which should be followed to increase our understanding, and I highlight this, to mitigate the risk. And the first one recommended was better characterization of the extremes. The second was to develop a more quantitative understanding of the processes which lead to these extremes. And what they argued was the third and most important thread was the study of impacts of these extremes on marine life. And I mean, this is all completely justifiable, in my opinion. As a Canadian, I'd like to also mention they forgot to mention sea ice, which is also an issue for significant parts of the ocean. But, when you look at that list of recommendations, you sort of ask the question, do any of these threads, recommended and important threads, any of them actually mitigate the risk? And I think, understanding is a prerequisite to mitigation, but nevertheless, should we actually do something about it? I'm going to be a little bit, and I'm not trying to be provocative, but to kind of raise some questions here. Now, Andreas has already mentioned the threat of deoxygenation on the global scale. There are papers published there which argue that at least with the high projections of future emissions, that this is a very major problem for oceanic life. A paper by Penn and Deutsch published a few years ago now argued that species losses from the combined effects of warming and oxygen depletion by the end of this century could be comparable to the other impacts of other human activities, whaling, fishing, habitat destruction, and even culminate in a mass extinction. Now, these are dramatic and slightly scary words, but within the realms of possibility if the high emission scenarios of CO2 emissions continue. So oxygen is a threat globally, for sure, but it's also, and it's maybe especially, a threat locally and regionally. And that's what I want to emphasize here, is that, if you look at the incidence of hypoxic events, which has been documented to be increasing, they're predominantly focused in coastal zones around the globe. And their causes are well known, as pointed out by Andreas. Certainly, excess nutrient inputs to coastal regions has been a major cause and continues to be a major cause. But increasingly, climate change is impacting these systems, including the coastal systems. So, for example, on the Pacific Northwest coast of America, the incidence of hypoxic events appears to be increasing. In the Baltic Sea, of course, a system studied for an awful long time and reasonably well understood. Again, despite efforts at nutrient reduction, the incidence of anoxic and hypoxic events appears to be increasing. And close to where I live in Atlantic Canada, and especially in the Gulf of St. Lawrence, it's been documented there's been a dramatic reduction of oxygen in the estuary and Gulf of St. Lawrence over the past, 70 years. Which appears to be attributable to large-scale reorganization of ocean currents, as well as some nutrient impacts, but increasingly the result of large-scale ocean circulation changes, which appear to have a climate-related cause. So, and this in the Gulf of St. Lawrence, but actually also in southwestern Nova Scotia, where I live, this is really a serious threat to coastal ecosystems and to commercial fisheries. So it's of considerable concern. So we've got these global scale effects, but we also have strong regional and local effects. And if we've thought about what's commonly called geoengineering, let's call it ocean-based climate interventions here, typically most of our discussion up until now has been focused on the mitigation of global climate change forcing, trying to do something using the ocean to impact global climate change caused by CO2 emissions. And that the pluses and minuses of that have been discussed greatly and have been mentioned in earlier talks this morning as well. But increasingly, we might be facing the need, as the inexorable climate change continues, and we've been relatively ineffective at reducing emissions so far, one has to say, that the mitigation of regional climate change impacts, such as those oxygen hypoxic events, might become increasingly important. But it's not just hypoxia, it's also sea ice loss. It has been mentioned already, local acidification events affecting coral reefs, and so on. So maybe the mitigation of regional climate change impacts is becoming increasingly important to consider. The energy transition itself, actually, in the case of oxygen, might actually offer us an opportunity here, and especially if during the during the energy transition, green hydrogen becomes a major component, then an advantage of green hydrogen production is that pretty much as a byproduct, pure oxygen is also produced, whichever form of electrolysis is used. Oxygen is a byproduct, and typically there's not really a market for that oxygen. In some cases there are, but very often there's no market for it, so it's just released to the atmosphere. And so actually 3 years ago, Science Day in Casablanca, I had the privilege of presenting there. And this kind of realization that green hydrogen production might actually provide a mitigation option was just coming into the forefront of our thinking. And this was one of the slides I presented there, and basically said we've got many, many questions. If we wanted to do this, could we pay for it using credits or offsets? What approaches would be effective and also responsible at the scale of the ocean? Would it be effective in mitigating biodiversity loss? And are there risks? A very important question. And then I kind of pointed out very little is known and that it's a new topic. And, 3 years later, say the questions are pretty similar. But as my colleague Patricia has pointed out, regions where green hydrogen is likely to be produced are also likely to be co-located with the regions threatened by hypoxia, because wind and prevalent in coastal regions and the access to transportation sites is likely where green hydrogen plants will be built. And the important points also are that even if, and we should, immediately reduce greenhouse gas emissions and also nutrient inputs to coastal regions, it is very likely, I think it's almost inevitable, that both warming and deoxygenation will continue for many decades to come. So the threat of hypoxia to coastal regions won't go away immediately. And so that raises an issue for the next 50 to 100 years at least, even if climate policies are enacted in the immediate future. So could green hydrogen production help mitigate hypoxia risk? I think some of these points I've made already, but I want to emphasize that large amounts of oxygen will be generated as a byproduct, large enough to address coastal and regional scale hypoxic problems, certainly not the global scale problem that Andreas mentioned. This is technically and financially feasible at scale. We sort of know that based on extensive experience from reoxygenation of large lakes and reservoirs. And that's where pure oxygen injection typically through various methods, but especially through diffuser technology, has been shown to work and has actually quite a lot of understanding in freshwater aquatic environments. But this has never been done at scale using this approach of injecting pure oxygen into the subsurface ocean. It's not yet been done in the marine environment. And there are different questions there, and there's different goals there. So a couple of years ago, Patricia and I wrote a kind of a thought piece there in Global Change Biology, where we pointed out that if you were thinking of doing this, and we thought there were good reasons to think about doing it for sure, it raises a whole bunch of questions cutting across all areas of science, social science, and policy, as well as natural science. And this is just a kind of a vision of the scale of questions that need to be posed and then addressed in order to consider whether to move forward in this. And I think the community right now is still defining these questions, recognizing they are significant and major. But as I say, right now, I don't think we've really moved very far in actually addressing them. Now, at the time, I think we said it was pretty urgent to address them because the pace of the energy transition was quite rapid, and especially there was a lot of excitement of building out green hydrogen plants globally. And we felt if we didn't start addressing them like straight away, we'd kind of miss the bus or miss the boat, and be too late to access the oxygen in order to use it. Now, the recent slowdown in the energy transition isn't a great thing, definitely not, but it actually might have bought us a little bit of time to conduct the necessary research. But this example I just took off the web yesterday actually shows what's happened, at least in North America, is where a very major green hydrogen project, which was proposed in New York State, has come to an end, and instead they're selling the land to be used as a data center. So I think the irony of that is not lost on all of us. But the pace of hydrogen plant construction, I think many believe, is likely to pick up again fairly soon. So I'm going to just summarize here, finish here, because leave room for questions. I think now, as 3 years ago in Casablanca, I think we feel that we urgently need to consider this possibility. Given the scale of the threat, especially to regional and local scale ecosystems in the marine environment. Likely what is needed is pilot scale marine demonstration projects, thinking up to maximum of 100 tons of oxygen per day delivery, conducted at different sites because no one location or ecosystem is likely to fit all. Different questions will arise in different ecosystems. And the reason we need this is really in order to be able to design, develop, and increase our understanding of the effectiveness of this approach, but also the risks, to develop and test monitoring and evaluation approaches, which will be critical to any implementation of this approach. And also related to the monitoring and evaluation come the need to be able to accurately model and also control the systems that could be put in place. So this all requires real field testing to evaluate how to do this before moving to any significant scale implementation. These projects would require, as the slide earlier showed, very broad transdisciplinary collaboration between engineers, oceanographers, ecologists, limnologists, policymakers, industry, local communities, indigenous peoples. It really is a very transdisciplinary problem. And it's the research at the pilot scale is necessary to deliver best practice guidelines and also help guide the design of governance frameworks which would be available to actually guide implementation. The risk is that we just rush into this without having done the pilot scale research, and I don't think anybody's feels very good about that idea. But we might have a little bit of time now to work together to evaluate this and think through our procedures using pilot scale projects. And I think that's my last slide. So hopefully that was clear, and more than willing to take any questions.
Thank you very much, Dr. Wallace, for the very interesting and impressive presentation. Okay, we have Flag Ocean Care.
Thank you for your presentation, Dr. Wallace. I wasn't quite clear when you briefly mentioned electrochemistry. I assume that's electrochemistry of seawater, but perhaps you can come back on that at the end when you respond to my broader question. As I understood it, you are trying to do two things with your proposal. One is to avoid or reduce marine impacts, negative marine impacts, and the other is to address the problem of climate change via the production of green hydrogen. When we look at electrochemistry at scale, which I presume this would be, we see that it would require substantial amounts of renewable energy, for one, and it would use and harm substantial amounts of seawater. So you're actually, in many respects, you're actually undermining the two things that you're trying to avoid. You are causing harm to the marine environment by processing large amounts of water, and you are inefficiently producing what you've referred to as green hydrogen, when that renewable energy that you're using to do that would be better and more efficiently used to offset the release of fossil fuel gases in the first instance, given that we're very much in a shortage of renewable energy at this point and are likely to remain so for the time being. And the other comment that I would make is that when you do electrochemistry, you don't just produce hydrogen and oxygen, you also would produce something like chlorine gas or hydrogen chloride, which is itself toxic and creates additional problems. So I'm not convinced that this is a legitimate climate solution, thank you, or local remedial solution, thanks.
Well, thanks for the questions. First of all, on the source of the water, right? And so at the moment, but this is changing rapidly. At the moment, many electrolysis systems rely on fresh water. And of course, that does have implications. You potentially, in many regions, you might well be competing for water with other uses and people and so on. And it has a whole array of environmental and social issues, which I think we're aware of, and I'm not going to go into right now. I'm actually not in the green hydrogen business myself, right? So this is not thought of from me. I'm kind of taking the assumption that this industry might move forward, and then what can you do with it if it happens? That's my viewpoint. Others, maybe have a different viewpoint. On the question of the use of renewable energy, I mean, I think I agree with your point. The renewable energy should be used to offset CO2 emissions, and that is the goal. But I think in this case, if hydrogen becomes part of the way to do that, green hydrogen, there is the potential to harness energy in more remote regions where maybe the energy demand is not so high, and use hydrogen either as a storage medium or as a transportation medium in the form of hydrogen, or maybe possibly other forms, in order to get it to places and markets where it's needed. So, that, again, I'm not got expertise in that, but that's not my field, but I do think there's also the issue of storage of energy when wind, for example, production is in excess of immediate requirements. Hydrogen becomes a potentially viable way to store it until that excess energy until it's needed. So there are kind of a number of arguments like that, which are plausible and need to be considered, but that's not really my point. My assumption was that, let's say this becomes a key part of the energy transition, then can we use the oxygen to do something useful and beneficial to the ocean with it? And there's lots of questions there as well. But that was where I was coming from. On the issue of the production of other materials, yes, and those need to be considered, especially especially if seawater is used. You may have desalination and so on, which raises additional issues. All of those aspects of the approach need to be thought through, but that again is at the front end of the question. I'm trying to say basically that if this moves forward as an environmentally acceptable technology, can we do something with the oxygen? And then there's a whole bunch of other questions. If that's okay, I mean, that's my viewpoint on it.
Okay, thank you. I see Germany, then Greenpeace.
Thank you, Dr. Wallace, for the presentation, and thanks for following up on this idea. I also recall your presentation you gave in Casablanca. I share a little bit of the concerns as to questioning this technology as such, as green hydrogen, and whether it's really a green technology, but that was already addressed. Further to this, I mean, I share all the sympathy for looking for wind solutions, and we all do this all the time, and we should pursue this pathway. But if we then look into bringing the oxygen to the ocean, I would think that this is a real massive human intervention that has a lot of concerns associated to this. I expect massive disturbance from bringing the infrastructure in to distribute the oxygen. We just heard about the sensitivity of digging out sediment and what it does to the carbon footprint. So you would have a lot of associated impacts. Then I would have questions to how it distributes. But I would like then also to make one aspect and whether you looked into this, how it would actually impact on the benthic ecosystems. So we need to mitigate especially oxygen depletion in the benthic ecosystems. And have you looked into the results we had from theoretical studies and some field studies in the Baltic Sea? Because this is an area where I've been working in a lot, where we wanted to use bottom oxygenation to combat eutrophication. And all the research that we've done so far showed that it would actually be not a good idea. And it is an intervention that is highly risky. For instance, if the pumps stop, we get huge pulses of toxic metals and nutrients coming out of the sediment, and we disturb a lot about of the bottom processes. So, yeah, I question whether you looked into this literature, and I know that the sentiment is a little bit different from combating eutrophication as to trying reoxygenation on a different scale. But I think there are still some similarities, and these experiences gained in the Baltic Sea could maybe be instrumental for thinking this a little bit further. And a second very quick question is, do you have any thoughts on who would pay for this? But because one aspect that in the Baltic Sea it failed and we didn't go to large-scale field experiments, it's hugely expensive, and yeah, that was also closing off the avenue. Thank you.
Thank you for those questions. And yes, certainly I am aware. I spent quite a little bit of time in Kiel, but I've been away for a while, right? But I am aware of, the considerable thought and thinking that's gone into this in the Baltic Sea. Now, my personal view, having then moved back to Canada, is that, I kind of realized that not all ocean systems are the same, and you're aware of that as well, right? So what we understand, based on the legacy of a long period of very high eutrophication in the Baltic region, that builds up a particular set of circumstances in the sediment and the enclosed basins of the Baltic, those lessons are relevant, but not necessarily the same as what would apply, for example, to the Gulf of St. Lawrence, where the hypoxia, it's partly due to eutrophication, but increasingly, really, the impact is due to circulation change. And and there's not so much legacy in the sediments yet now, because the things are happening differently. So that's the point, is really not, I don't think one size fits all, and learning in one system like the Baltic, of which we know quite a lot, doesn't necessarily apply to other parts of the world where the circumstances might be different. And I think there is a risk to kind of thinking that way and then not considering that the questions might be different in other regions. I know that sounds a bit vague, but we have thought that through a little bit. And that's why I still think pilot scale experiments in different diverse sites may well help us to understand that better, okay? In terms of expense and who would pay for it, yes, I mean, you mentioned disturbance. That's definitely a consideration that has to go through. But again, we are talking about ecosystems which are under severe threat anyway from a problem. And then there is a moral and ethical issue to address whether, and I think we raised that 3 years ago, whether we let it happen, or if we can do something, we should do it anyway, recognizing nothing we do in the ocean is without an impact, right? So there is a balance there that I think, again, pilot scale research can help us evaluate better. But the impacts are coming. The impacts in many systems are coming, and therefore, I think, personally, think we need to at least consider thinking about them. In terms of who pays for it, excellent question. And again, compared to, I think, a lot of the research into marine carbon dioxide removal to address a small part of a global problem is going on because people can make money out of it. It's as pure as simple as that. Here, with this approach, the motivation would have to be different because at the moment, I don't think there is a mechanism for intervening in the ocean environment to protect it, right? Which is interesting in and of its own right. So there's not a clear financial mechanism. Possibly some companies could get some credits, but the system I don't think is in place at the moment, and may need to be developed as part of the research into this possibility. That's my feeling anyway.
Thank you. We will have one more question by Greenpeace.
Thank you, Chair. And yeah, thank you very much for that interesting presentation. Just coming back to two questions. So one, presumably, if you are going to be dosing oxygen close to the seabed, you need to get the dosing right so that you've got some idea of predictability of what you're doing. So could you say anything about the complexity and scale of the monitoring you would need to be doing to make sure that you could understand and monitor the oxygen levels and their changes at the seabed? I mean, what kind of monitoring network would you need in order to make sure you're not overdosing or getting very variable dosing at the seabed? And secondly, I was interested that in response to one of the early questions, you said that you're not in the business of green hydrogen production, which confused me a little bit because on the first slide you had the logo of Life, which is a company that produces green hydrogen. So maybe this question isn't for you and maybe for your co-author, but on the Life website, it initially says that it produces hydrogen with 0 CO2 emissions. But when you look elsewhere on the website, there's quite a different story. I mean, there is still CO2 emissions from producing the hydrogen. So but that may not be a question for you, but maybe something to take back to your co-author. Thank you.
Yeah, I should have clarified that. Thank you for that. I mean, I'm Patricia's colleague and somebody I've worked with. We do not share funding, for example, and things like that. But we do discuss this problem together from diverse perspectives, probably share some opinions for sure. But Life, of course, is a commercial entity that is interested in hydrogen production, is doing it, and interested in this possibility of taking the oxygen and using it for reoxygenation. So I probably should have clarified that a little bit in my answer earlier. But nevertheless, on the other aspect, on the monitoring, that's an excellent question. There's no immediate answer to that. I think we have technologies which could be applied for this problem, mobile and fixed sensing technologies. We'd need to focus much more on nearshore modeling, near field modeling. Higher resolution and near field, because the near field effects might be very significant. There is experience of this in freshwater environments, but they're not the same as marine environments, obviously, with tides and circulation patterns and so on. So this would be a very, very key part of any pilot study, would be to work through the technologies needed and the modeling approaches needed for actually any possible implementation. And it's probably a long time. I'm trying to think. It's probably a long time since the marine science community has tackled a problem at this scale. I actually can't think of one. So it does push us into a kind of a new application of ocean monitoring. It's not like Argo floats in the global ocean or the large scale models that Andreas was using, for example. It's much more focused near smaller scale approaches. So it is, I think, fundamentally different than a lot of things we've done up until now in the oceanographic community. But I wouldn't say that it's not something out of line of what maybe other communities have done. Is that a reasonable answer to your question? I mean, I think it is a new area. That was of research that we're talking about here.
Thanks very much.
Okay, thank you again, Dr. Wallace, for your time early in Canada. And our final presentation today is by Dr. Catherine Romanak. Dr. Romanak is a research professor at the Bureau of Economic Geology, the University of Texas at Austin. She is a leading expert in carbon capture, storage, monitoring, and risk assessment, with nearly 2 decades of experience in geologic CO2 storage research. Her work focuses on near-surface monitoring, groundwater protection, and environmental assurance. The title of her presentation is Climate-Driven Ocean Variability and the Challenge of Monitoring, Lessons from a CCS Case Study. Dr. Romanak, you can start when you are ready.
Hey, you've unmuted me. Thank you. Thank you so much for that introduction. Yes, my name is Dr. Catherine Romanak, and I'm very pleased to be here today to talk to you about how climate-driven variability in the oceans is impacting monitoring of offshore CCS storage sites. And I want to start off by saying I am not an oceanographer. I started my career as a soil gas geochemist working on onshore projects. And I spent a lot of time trying to figure out how we would distinguish a leak of CO2 in the soils from natural variability. And then over the past 10 years, as the United States has started to move into the offshore realm and looking at offshore CCS, I became very interested in how can we take the lessons and the challenges from onshore and how we can apply those in the offshore. And so I want to apologize up front that many of the slides that I'm going to present might have land above the storage site, but all of the concepts that I'm going to be talking today are also applicable in the offshore. And one of the things that I immediately saw was that any variability that you have onshore is like 10 times more in the offshore. So I think there's a lot of great lessons that we can learn here. And I'm going to take you through those lessons. And then at the very end, I'm going to show you how they apply to offshore monitoring of CO2 geological storage sites. But I think there's one very subtle but powerful thing that it's an observation that I need to start with. And I think we all need to start with this. It's subtle but powerful. And that is that when we monitor for pollution in the offshore, we know when something shouldn't be there. These are toxic chemicals that are not really naturally present in the offshore. And so in this case, concentration thresholds really matter and they work. But what we're doing in CO2 storage monitoring is we are monitoring for a component that is already naturally abundant in the environment, and it's cycling all over the place, and it's being impacted by the variability of the environment. So I think just keeping that in mind as we talk about how we're going to monitor these sites in the offshore is really important. I think we all know that the storage regulations were given in the IPCC greenhouse gas guidelines. Those are the backbones of global regulations and policies, and it's very well developed. And these regulations set out the actual monitoring activities that we need to do. And we do these monitoring activities all throughout the stratigraphy. And again, apologies that there's trees on the top of this diagram, but you can also imagine the ocean on top of this diagram. And what I want you to see here is that what I'm going to be focusing on is the biosphere, or the seawater column, and the marine floor, marine sediments, because this is the interface between the CO2 being in the subsurface versus the CO2 being re-entering the atmosphere. And the one thing I want you to get from this is that in the deep subsurface, it's very quiet. There's no variability at all. The variability that we see comes from the actual injection of CO2. But as we move up into the environment, which is the environment that we want to monitor, we see so much variability, and we need to deal with that in our monitoring. We also monitor during all the phases of the project. This begins with the baseline and baselining and site selection and getting the permit, through operations and closure and the post-closure phase. This really has implications for the impact of climate change on monitoring, because monitoring in many regulatory schemes needs to happen for over 100 years. So you can just imagine at the beginning of a project, trying to understand how you're going to predict leakage over that long time span when we have climate change happening. So that's really the first impact of climate change on CO2 storage monitoring. So these regulations say we're to go out and look for leakage or anomalies, and we're very good at that. We have so many different ways on land and in the ocean that we can go out and look for CO2 anomalies. But the question is, what are we actually finding when we look for those anomalies? Well, we're actually just finding areas that have a lot of CO2 and areas that don't have so much CO2. So what we need to do is understand what is the cause of those anomalies, and that is called source attribution. Now, just an example, I have two onshore examples. The one on the left is at the Weyburn CO2 storage and monitoring site. This is a monitoring grid, and here we see areas where there's blue elevated CO2 and areas where we do not have elevated CO2. This CO2 is being produced in situ in the soils by microbial respiration. But on the right-hand side, this is a volcanic area where CO2 is coming from the bottom, coming from deep and coming up to the surface. It's a different mechanism, but the CO2, it looks the same on the surface. These volcanic analogs are very, very similar to if we would get leakage from a storage site. But again, on the surface, they don't look any different. How do we know which mechanism created that anomaly? Well, the regulations put forth that we use baselines because, again, this has been the traditional pollution monitoring is baselines. So we know, and it's very well understood in CCS, that there is seasonal variation to CO2. So we need at least 1 year of baseline to understand seasonal variability. And then we monitor the CO2 either on the seafloor or in the water column. And any CO2 concentration above baseline indicates an anomaly. But it actually indicates an anomaly, and then we have to go in and ask the question, did the project cause the anomaly, or is it just variability? And this is called source attribution. But I think we can all, I think we can all understand that if we are using baselines to detect anomalies, then what's going to happen in a changing climate over these long time periods? We would get a false positive for leakage. And that is because we know that baselines in soils are shifting upwards because of climate change. We know baselines in groundwater are shifting upwards because of climate change. And we all know that baselines in seawater are shifting upwards because the oceans are absorbing the atmospheric CO2. So again, what's going to happen if we use baselines for attribution and then we monitor over many years, we will get false positives. And in fact, we were starting to think about that in the terrestrial area when it actually happened at a project. So this is the Tamakamai project. It's an offshore demonstration project in Japan, and they did everything right. They did everything according to the regulations. They derived their leakage thresholds from in the seawater column from 1 year of baseline data, and then they began injecting and they began doing routine environmental monitoring, and it only took 1 month for them to find a seawater CO2 concentration that was above the threshold. So the project was shut down for almost a year while they tried to work out whether that was leakage or not. And in the end, they realized this was not leakage at all. This was variability. So if we use baselines, they are shifting and they will cause false positives and project shutdowns. And we all know that we have to balance the protection of the ecosystem from these projects. Okay, but we also need projects that are operating correctly to be able to continue to operate because we've got to get the CO2, stop putting it in the atmosphere. So this is a balance, right, between making sure that projects move forward, but also protecting the environment from any leakage that might happen. So we were very aware of this, and we decided we need to do something about this, and we needed to create a new approach, not baseline. And so we developed a process-based soil gas ratio approach that uses the simple ratios of the coexisting gases to understand the processes that create the gases as they are. And these incorporate all the different processes that actually create variability. You do not need background baseline methods, and it can be applied in any environment. So I'm going to quickly walk you through some of these very simple but powerful relationships so you can understand it. And then I'm going to show you how it's being applied to marine monitoring. So if you have nothing going on in soil, you have atmosphere, which is 21% oxygen and very little CO2. And as biological respiration happens, 1 mole of oxygen is consumed for every mole of CO2 produced, and you are basically moving up and down this respiration line, okay? So keep note of that line. That line is important. We can also look at other things like methane oxidation. I won't go into that here. But what we also know is that anything to the left of the line, of the respiration line, means that some water has infiltrated into the soils, grabbed the CO2, and taken that out, okay? But if we get CO2 to the right of that line, then that implies that CO2 has come from outside the system, and we may have leakage. There's another ratio we can use, nitrogen. Nitrogen, at least in the soils, can be seen as a tracer. It really doesn't change very much. We don't see much change. So we can use it as a tracer in the gas by the fact that we look at the gas ratios in volume percent. If we take CO2 out of the pore through dissolution, nitrogen goes above atmospheric, and we have a natural signal. However, if leakage comes into the pore, it will reduce all of the other volume percents of the gases, and nitrogen will be depleted relative to oxygen. And so now we see that we have several indicators of leakage by looking at these ratios. It is simple, it is transparent, it is graphical. The answer is immediate. And it's universal. It can be done anywhere. And what's beautiful is if we're looking at these long time periods, long-term variability is inherently included in the method. Respiration, when we have hot and cold, just moves along this line. And when we have wet and dry, it moves right and left. So this is a beautiful thing. One other thing I want to the reason why it's very important to have these simple methods is for stakeholder engagement. Now, I know we have more stakeholder engagement on shore than we do offshore, but please realize there are stakeholders in the offshore, shrimpers and fishermen and oysters, all types of things. So it's very important to be able to communicate and be transparent about these methods. We did have a leakage allegation from a landowner living near a project where they looked out and they saw changes in their land and they wanted to know, is this leakage? We see these weird things happening in our land. And so they had a consultant that went out that was inexperienced in attribution, and they said, yes, it's leaking. And so all of this sensationalized media went out about how this is leaking. But we were one of the teams that were called in, and we used a process-based approach. And the beauty was we could talk to the landowner. They followed us across the land. We told them the rules of the game up front. It was very transparent. They could see with their own eyes whether there was leakage or not. And in the end, they were actually convinced by our study because it included them and because they could understand it. So this is very important part of this. And what I also want to bring out is that this is another way that climate change impacts our monitoring, because landowners and fishermen and shrimpers are going to start seeing the impacts of climate change, and they're going to ask if it's because of leakage or not. So this is really important that we get these approaches right. So if I go back to my first example, this was actually the project on the left where the Kerr farm was, where the landowners were. And this is the actual data set that I collected with the landowners walking alongside me. As you can see, as we went across the CO2 anomalies, we saw they were natural signals. They were on the line or to the left of the line. But on the right-hand side is a controlled release experiment where we put CO2 into the environment on purpose to try to understand what it would look like. And we do see, indeed and in fact, that a leakage signal manifests to the right of that line. Okay, awesome. So we've been presenting this at workshops, and the Tamakamai project picked it up, and they used it at Tamakamai. So we're going to talk more about that false positive at Tamakamai. But also we have our Norwegian colleagues who have also been developing these methods and trying different approaches. Okay, so this is the data set at, this is Osaka Bay data, and this is 10 years of Osaka Bay data. And what these scientists saw was, okay, there is a trend. The process-based trend exists in the ocean. Here it is. It's the respiration line, basically. But what you see is there's so much scatter in the offshore that they still had to create thresholds, okay? Here are the actual monitoring data from Tamakamai. Here you see in the gray are the baseline data that they collected. So the dark ones here are the baseline data. And then the blue are the monitoring data. And then the orange is where the monitoring data went over the threshold, right? So what we see here is that the variability in the offshore is so much that it could not only stop us from seeing a small leak, but it's also very difficult for attribution. Here is what I want you to see. This is the 10 years of Osaka Bay data. Now I'm going to overlay 1 year of Tamakamai data on this trend. And there it is. So what we see is that the temporal variability, even under climate change, is so large that the scatter makes monitoring very difficult to be able to attribute these signals, right? Every time you make a measurement, just about, you're going to get a different answer. So what we really need to do, and what we're working on doing, and I'm working with Abdir Omar at Norse, he has a method called CSEEP, and what we're trying to do is systematically normalize these key parameters by extracting the variability of all of these different processes. And these are the data from the STEM CCS controlled release. So it's a short-term monitoring, but what we see here is a reduction in the scatter by normalizing these parameters. And then this black here is the actual controlled release. So now we're starting to see some progress on being able to very cleanly and clearly see a leakage signal in the offshore. So the conclusions are that, first of all, remember that CO2 monitoring is very different because CO2 is already in the environment and it's cycling between all different types of parameters. And as atmospheric CO2 increases and environmental systems respond, background concentrations are rising and natural variability is increasing. So the long-term CO2 uptake is actually making baselines ineffective. And then the environmental change from the more extreme weather events are creating scatter in the data. So we don't just need to find the leakage, but we need to attribute it. And these process-based approaches are being developed, and they're distinguishing leakage from environmental variability over long time sequences without the need for baseline. And they inherently include these natural variations for long-term monitoring. And what we are seeing is that right now in the regulations, attribution is not emphasized, but more and more as these regulations are updated with scientific advancements, they are increasingly being incorporated into the regulations and guidance of CO2 storage. So thank you very much for your attention, and I'm happy to take questions.
Thank you, Dr. Romanak, and sorry for keeping you waiting for a long time due to delay.
It's okay.
I think we have time for one or two questions. Oh, Italy. Sorry.
Yes, thank you very much, and thank you very much for the presentation. It was very interesting. Please bear with me, I'm not an expert in this field, so I hope my question makes sense. I noticed that in one of your slides you mentioned the stable isotope analysis. So I'm wondering if the stable isotope analysis of carbon and oxygen can give a more precise response, and if so, if you used it to validate your methods. Thank you very much.
Does make a lot of sense, and it's actually a great question. Yes, isotopes we've looked at a lot, and what we have found, unfortunately, is that many times they overlap. And in fact, the overlap of isotopes between what was in the soil and what was being injected at Weyburn, at the Kerr farm, is the reason why the consultant thought it was leakage, okay? So we do know that there's a lot of overlap in the isotopes right now. And we're also looking at hub models where right now many different sources of CO2 may go into one storage site. And so so definitely we do use isotopes, but there is a limited application of them. I think it may be more it might be a more stronger tool in the offshore, perhaps. But right now, there's so much overlap that we really can't just solely think about isotopes. And then also, the isotopes take time to get the data, which is fine. That doesn't matter. But it is we have found that it's very useful to have these stakeholder-friendly approaches so that we can bring people alongside us and we can help them to understand these systems and feel safer. So thank you that's a great question.
Okay, thank you. I see no more flags. Okay, thank you again, Dr. Romanak, for the great presentation. Thank you. Now we have reached the end of our time. So as I proposed, we tried not to register Q&A time. As a result, we do not have time for the discussion session. So it is now time to conclude our symposium. Before we do that, I would like to invite the chair to provide the final closing remark.
Thank you, Vice Chair and the current chair of the Science Day, and distinguished delegates, colleagues, and all attending remotely this Science Day. It's been a pleasure on behalf of the scientific group to have the chance to deliver this Science Day symposium on the effect of climate change on permitting monitoring under the London Convention London Protocol. The topic was chosen in its broad meaning, including many climate-related issues to ongoing research findings affecting the marine environment. Among those, as we saw in the previous presentation, the effect of increasing CO2 combined with global warming, the variation of oxygen content in the marine environment, flux of sediment and organic carbon, and finally, as we saw in the last presentation, the monitoring of the CCS and affecting its variability. I believe that all the presentation delivered, despite not directly linked to the permitting as the title, but there was a monitoring implication, were really scientific relevant to open the way toward an effective implementation of the London Protocol in addressing climate change issues. As the chair of the scientific group of the London Convention London Protocol, I wish to thank all the speakers who provided a remarkable contribution to this Science Day. I wish to express my gratitude to the Vice Chair, Dr. Yon Chang, for his excellent dedication and work in the organization and chairing in this Science Day. You succeed very well again, and I congratulate you. I express my warm greeting and appreciation to the first Vice Chair, Ms. Jessica Mans, for her invaluable charisma and availability to support her maternity big journey. I also wish to extend my best thanks to the LCLP Secretariat, Dr. Andy Bechner and Mr. Frederick Hag, and the IMO staff for the valued support to the Vice Chair. A special mention to Ms. JP here, who has been so helpful, focused, and competent in the preparation support to this symposium. I remind that all the presentation given today will be made available in the dedicated webpage to the Science Day at the IMO website at the previous Science Day, as well as the YouTube stream, in order to be accessed and spread over a wider audience. Finally, I want to highlight the importance of the Science Day to provide insight and contribution on important topics chosen by the parties of LCLP in a more informal pencil-door setting. And the resume of face-to-face meeting is preferable to enhance interaction, but as well the hybrid capabilities and the online streaming provided a wider audience and involvement of speakers across the globe. And last but not least, students and other interest stakeholders. Among those, I want to mention the students of the Graduate School of London Protocol Engineering Master of Project Administration, the LPM, from the kiosk that were invited to attend this meeting remotely. So I wish to thank again you all, and looking forward to have next year Science Day on a topic of relevance to be discussed and decided early. I give the floor back to you, Vice Chair.
Thank you, Chair. And thank you once again to all of our distinguished speakers and to all participants here and online. And with that, I would say the 2026 Science Day is closed. Thank you very much.